allows us to do targeted genomics using light switch. You can imagine it's like shining a spotlight on the cell of your interest, and which allows us to do further analysis based on the mark that you make on those cells. Genomics is the science that analyzes the sequence or function or evolution or structure of the genome of the cell, right? So people have been doing genomics for a few decades now. This includes not only analyzing the sequence of the DNA in the cell, but also the RNA molecules in a cell, in which you can know what is the expression pattern, what kind of RNA molecule is enriched in different cell types, etc. So the genomics is very useful for a lot of different purposes. In the early days, when we do, to try to proceed this same, yeah. So in the early days, when we do sequencing, we do what is now being called a bulk sequencing, which means that you have a group of cells, you wash them together, you lyse them together, which means that the genomic DNA or mRNA molecules from different cells will mix with each other, and then you send the whole thing for sequencing, right? So whatever you get from the sequencing experiment is the average of the population. Okay, that is very helpful, and that is already very amazing. But nowadays, scientists has been developing and using what is even more precise, which is the single-cell genomic analysis. And the single-cell analysis enables the studies of different things that you won't be able to see if you were just looking at the average in the bulk sequencing. These include the low incidence events. For example, if you are studying something that only happens in 1 to 2% of the cells, it's a rare event. In that case, if you only look at the average result of a bulk sequencing from a population, you will never be able to see what you wanted to see, right? So you need single-cell sequencing. Another example is that if you're interested in cell-to-cell variation within a population, you cannot study that just by looking at the average of the population. You need to look at each individual cells within this population to know the variation, say in gene expression. In the last example is that in a tissue, it's often heterogeneous, right? So in a tissue, there will be different cell types, and some of the cell types is going to be really rare. In that case, if you analyze the whole tissue, you won't see those rare populations. And you're going to need single-cell analysis, which allows you to isolate and analyze the cells of your interest. All right, and this is not the focus of today's class, but hope you will understand that going from the bulk sequencing to single-cell sequencing, the difference is that you have to change from you get a lot of DNA or mRNAs from many cells to you're trying to isolate one cell and trying to amplify the DNA or mRNA from that one cell. And the amplification needs to be precise. You cannot introduce too much bias in this process. And so this is, so single-cell analysis is, it has been developed for many years, and it's now a relatively mature technique, and more labs are using. As you may imagine, in single-cell analysis, the key to success is how you're going to identify and make a mark on the cell that you want to analyze, right? So you don't want to mark the wrong cell and then end up analyzing something totally different, or you don't want to miss the rare population that you're really interested in. So scientists has also been developing methods that allows you to do targeted single-cell analysis. One of the workflows, so I'm listing two different possible workflows. The first one is that you can physically separate the cell from the beginning of your experiment, which means that you would use single-cell sorting and sort individual cells into 96 well or 384 well plates. So every single is like they live in a gel like well without any companion. Okay, so you can physically separate the cells. You just do a lot of microscopy to observe those cells. And if you're studying, say, a rare event, and once you observe, oh, now this weird cell division happened in this particular cell in this well, then you would only wash out those cells or lyse that cell in that well and then analyze the sequence, either the DNA sequence or the mRNA abundance in that particular cell, right? So in this method, you would separate the cell first, followed by huge observation, and then do sequencing. This is a method, one of the examples of this type of method is the Look-Seek experiment that's developed by the Pellman lab, which is the lab that I did my postdoc research at. And this is the topic of the paper that's going to be presented in the student's presentation in the next hour. But we're going to spend this hour talking about a different type of possible workflow, which is basically you flip the first two steps and you start with the observation of the cells. So similarly, you do imaging. After imaging, you will pick the cell that you want to further analyze by sequencing, and you separate them out. So it's different from the first workflow because you start the experiment with a fairly intact, say, piece of tissue or multicellular like sample, right? So this is going to be more physiological relevant than if you have to physically separate the cell into wells in the beginning. So the left workflow, the method, is kind of limited to do cell biology, so you can use it to observe rare events, but if you're more interested in something that only happens in a tissue or something that relies in cell-to-cell communication, then the blue workflow won't work for you. Okay, but in the green workflow, you observe the samples first with the least perturbation, but then you need a different method, trying to mark the cell of your interest and trying to separate them out, right? And this is what we're going to talk about in this lecture. And the principle is actually quite easy. So usually people would use a targeted illumination followed by FACS sorting to single out the cell that they want to do single cell analysis. And again, this can be either whole genome sequencing of the DNA sequence or the single cell RNA sequencing and analysis. So the idea is that, say, in the field of view, there are different types of cells or different groups of cells. And after some observation, you decided, oh, okay, it's this circled area that I want to do further study on, okay? So you can do targeted illumination to make these cells now glow in green. Okay, so if these cells are the only cell that is glowing in green in the whole population, then you can use the fluorescence activated cell sorting or FACS to separate these cells. Either this is a single cell or a group of cells. Either way, it's going to work. You can use FACS sorting and say, okay, I only want to select and sort, isolate the cells that has a high value for green fluorescence. Right, so in that sorting, you're going to throw away all these other cells, but these cells are going to be retained in the tube, and you can do single cell analysis after this sorting. Okay, that's the principle, and that's the end of what I'm going to talk about. No, that's a joke. So this is the principle which is, related to everything that I'm going to talk about in the next hour, but hopefully I will be able to convince you that even with this simple principle, there can come with many creativity, and scientists can use this simple principle to design many different methods to address different type of questions. And hopefully you will be able to appreciate the creativity behind all different kinds of methods, which is the purpose of this class anyway. Okay, so in order to do targeted illumination, we need a special kind of chlorophyll, basically. right? So this can be done by either a photoactivable fluorescent protein, which means that they are not fluorescent in the beginning, but once you shine a specific wavelength of light on those fluorophores, usually UV or blue light, they will start to emit fluorescent signal. For example, the PA-GFP is a GFP variant that won't glow if you just shine a normal light on them. But if you shine a specific wavelength of blue light or UV light, then you will start to receive GFP's fluorescence. Similarly, the PAM cherry would give you a red fluorescence if you activate those. On the other hand, there is a group of fluorescent protein that's photoconvertible, meaning that they already are fluorescent even before you activate or convert the fluorescence. So these are Dendra or EOSFP, and there are other types of photoconvertible fluorescent proteins. And each type of proteins that I talk about here, there are scientists that's devoted to like further optimizing how well you can convert the signal, how bright is the signal, etc. So you're like in some literatures, you're gonna see, say for example, Dendra, Dendra 2, EOSFP 2, 3, 3.2, etc. Okay, so Dendra and EOS, they usually like before activation, they would give you green fluorescence. But if you shine a blue light or UV light on them, they will switch to emit red fluorescence. So the change of color is easy to visualize, and also the change of color allows you to now use the converted wavelengths to isolate the cell of interest in your experiment. And of course, we're interested in single cell analysis, right? So the scientists would like to know whether they can actually photoactivate or photoconvert only the cell of their interest. So in this figure, they show that yes, you can do that. And you can do that not only for human cells, but also for smaller cells like yeast cells or bacteria cells. And in these cells, in addition to the PAM cherry, they also express the GFP in the cell. So then, even without photoactivation, you can visualize the location of each cell in the field of view. And the arrow indicates where the activation is going to be. Say, okay, after observing thousands of cells, I finally find that this yeast cell has something that I want to further analyze by sequencing, right? So I shine a light on these cells, and nice to show here is that you will see a very strong red fluorescence from where the target cell is. And even more excitingly is that you will see that even though, especially in this field of yeast cells, even though it's crowded, but you don't actually, photoconvert or photoactivate signal from the neighboring cells, which means that the control is pretty excise. So you can do single cell optical tagging by this method without any problem, and this is has been used in different purposes, which I'll show you in a second. But as you know, say if I have many field of view like this, and we usually do live cell imaging to observe the samples, right? So if I have to just look at all the cells and pick which one I want to photoconvert, it may take a long time, right? And human eyes are, it's easy for me to miss things if I just rely on my eyes, and I may not be able to screen through that many cells anyway. So scientists have also tried to develop more, methods that uses artificial intelligence to help with this process. In this paper that's published in 2021 in Journal of Cell Biology, this group of scientists tried to implement the AI system into the targeted illumination or targeted genomics. And they first want to test if the AI system they use is going to be able to detect known mixed population. So the method they use to test their system is that they have many cells that all express a GFP parkin. So parkin is a protein that is associated with Parkinson's disease. It's normally in the cytosol, so you can see the green signal in the cytosol in every cell. But parkin is known to translocate into the mitochondria when the mitochondria of those cells are dysfunctional, and it's about to undergo mitophagy, which is a process that allows you to degrade the sick or dysfunctional mitochondria in the cell. Okay, so if in the population of cells that all express GFP parkin, mitophagy is induced, then the parkin would translocate into mitochondria. But this translocation depends on another protein called PINK1. So if you mix in a small population of PINK1 depleted cells in this population, then you're going to see these two cells, they won't have translocation of parkin, right? And so the scientists use this system where they purposefully mix in some SG PINK1 cells into the whole population and see if their AI system can detect those cells that doesn't have a mitochondrial parkin. If the system can detect accurately those ones, they would photoactivate the cells, and then they would study what they isolated from this method. And the result is like that. So here they try to mix in the PINK1 depleted cells in 4 different ratios. So what is listed above the figure is what they mixed in. And then what is shown here is the FACS data, with the top right corner being the ones that they wanted, or the ones that sorted after photoactivation. And the percentage here reflects how much they isolate, how many cells they isolated after photoactivation. So what you will notice is that the capture is not 100%, so the AI didn't capture, say, 100% of the cell that it should capture, but it could capture about 70–80%. And also, even when you only mix in a fairly small population of the cells with translocation defects, the AI can still catch them. So the authors decided that, okay, using this AI photo switchable screening method, they could potentially use this in actual screens to identify new and interesting factors for the translocation of different proteins. So in the same paper, what they're actually interested in is to design a CRISPR screen to isolate factors that's required for the translocation of a transcription factor called TFAB. So TFAB is a transcription factor that is going to be translocated into the nucleus. You'll see the nuclear green signal here if the cells has been starved. Okay, so upon starvation, TFAB would translocate into the nucleus. But this translocation is not permanent, and after the cells activate a different pathway, the TFAB signal would be going back to the cytosol again. But this cytosolic second transportation of TFAB is not well understood. So the scientists wanted to know what is controlling this process. So they designed a screen where they transfected the TFAB GFP cells with a group of a library of lentiviral sgRNA library, and then they select for the cells that has a nuclear TFAB after a long starvation, when other cells has already has cytosolic TFAB signal. So they use the same AI system as I mentioned in the last slide to identify these cells with a nuclear TFAB GFP, and then they can photoactivate this one and use the FACS sorting to isolate these cells and then analyze them by next generation sequencing. So this is approximately how it looks like. So in the top left panel is the TFAB GFP signal. And in order for the AI program to accurately describe or segment the nuclear area versus the cytosol, they also introduced a nuclear marker. So it took about 1 second for the microscope to collect the image, and it took only about 5 seconds for the AI to automatically segment the image and then define where is the nucleus versus where is the cytosol. And they can also calculate how much TFAB signal is in the nucleus versus in the cytosol for you. So after 5 seconds, the program can already identify this cell with the nuclear TFAB signal, and you would then photoactivate this cell. And you'll see that before photoactivation, there's no PAM cherry signal in the same field, but after activation, only this cell is lit up. And then they can use FACS to isolate the cell that has the high M cherry signal and analyze them further. So in this screen, the authors screened a lot of cells using this AI-based program, and in this particular example, they isolated about 0.5% of all the cells that they passed their threshold to be the cell that have a nuclear TFAB signal. So using this method, they identified interesting and previously unknown factors that is specifically required for the retranslocation of the TFAB transcription factor. And we won't talk about too much results today, but I just hopefully you can appreciate that this method not only can help you quickly identify your target, but it's quickly identifying the translocation or a different cellular localization of the protein of interest too, which is a very good result. Now, a similar method can be applied to study the spatial heterogeneity in a tissue. What I'm showing you here is 2 different mouse lymph nodes. So the lymph node is our immune organ, right? And then in the lymph node, there is some heterogeneity that people are really interested in. It was reported previously that near the inner part of the lymph node, there's more T cell, or it's called the T area. On the other hand, near the outside of the lymph node there are more B cells, or it's called the B follicle region, or the B cell niche. But scientists are still interested in knowing what is the difference between, say, the T area and the B follicle, and are there any rare population of cells there, what kind of markers these cells are expressing, etc. So what they did is that in a transgenic mouse that's expressing PAGFP, they inject the mouse with fluorescent T or B N or B cells. So this is they are using the T and B cells to roughly mark the spatial heterogeneity, different regions in the lymph node. So once they mark that, they can selectively photoactivate either the cells that's in the T area or the cell that's in the B follicle, and then they would isolate these cells and analyze what is being expressed and what type of cells these may be. Right. So this method is very useful in terms of looking at heterogeneity in a tissue that is hard to define what is the inner component of these tissues. Okay, even more creatively, there is another group of scientists who's using the same method, but instead of studying cellular biology or looking at the spatial distribution of different cells, they are actually using this method to isolate or screen for a better fluorescent protein. So if you have studied or if you have used yellow fluorescent protein, you may already know that the yellow fluorescent protein is very photo unstable. So if you shine light on a YFP, it will photo bleach very quickly. And this has been some struggle of the scientists, is that we couldn't find a very photostable yellow fluorescent protein yet. So the authors of this paper wanted to address that. by designing a screen. So what they did is that they transformed the E cells with a library of the construct that has a different variant of a yellow fluorescent protein called M. venus. So this same construct also carries PAM cherry and tag BFP, which is used to identify each individual cells. After transformation, so the E cells within the transformed population would express a different variant of the yellow fluorescent protein. And then the scientists would use would do a photo bleaching experiment where they would photo bleach the yellow fluorescent protein in these E cells. So after 45 seconds of photo bleaching, they would select for the E cell that is still emitting the bright yellow light, and then target that, take that cell, and then undergo single cell sorting, analyzing what is being expressed in that particular E cells, because that could be a better yellow fluorescent protein that everybody is looking for, right? And the image on the right shows how it looks like during the experimentation, which is that you have thousands of E cells, each expressing a slightly different version of YP. And before and after photo bleaching, you're going to see that after photo bleaching, you do see dimmer signals of the YP channel. Of course, by eyes, we won't know which ones are more stable, but again, AI is really helpful in these processes. And the figure below shows that you can compare the brightness of YP to the BFP that's used to identify the cells. You can also compare the fluorescent intensity before and after photo bleaching to estimate the photo stability of each YP variant. For this purpose, the authors wanted only the cells in the red group, which is in the top right corner, because these are the brighter signals and has higher photo stability. And using this method, they isolated a group of E cells and find a bunch of different YP variants. What they found is a specific variant of YP, which they named M gold. And they compared the brightness versus the photostability of M gold in yeast and human cells. And they found that even though M gold is not brighter than other YP molecules that scientists have been using, but they found that M gold is significantly more photostable than any other widely used YP fluorescent proteins. So using this method, they found the most photostable YP reported today, which demonstrated how useful the targeted illumination can be used in a screening like that. Okay, so far all the methods that I told you relies on transgenic animals or transgenic cells, right? And this has to be pre-engineered, which means that before you started all this experiment, those animals or cells or yeast has already they already expressed the photoactivable GFP or M cherry. You just didn't see the signal before the activation, right? And for people who work in a lab, you will know that that's not always the case. It's not always easy to design the experiment where your transgenic animals already have the photoactivable version of a fluorescent protein, right? Because you may need the channel you may need a fluorescent channel to image something else. Sometimes the transgenic won't work, right? All kinds of possible technical issues. To address that, there are different kinds of methods that doesn't rely on pre-engineered transgenic cells or animals, which I think is also amazing. So these are the things that we talk about, and now I add a question mark here. So does targeted illumination actually requires a photo-convertible pre-engineered protein? The answer is no. There are other possibilities. For example, scientists have been using photoactivable dye. It's just a dye, so your tissue is not pre-engineered. You just add a dye, and you can photoactivate that. We'll show you an example in a second. It could be also that you can use a photoactivable chemical reaction which allows you to mark the cell of interest for your purpose. So some of you may be familiar with a series of dye called JF something something something. So these are developed at Genelia Farm, which is affiliated with Howard Hughes Medical Institution. So they develop a series of dyes that's bright and stable, and you can use them for all kinds of purpose. So that's a JF dyes. 549 stands for the wavelengths in which the dye can be activated, right? And the JF dyes also comes with the photoactivable version. So if you use the photoactivable dye, it's just like a photoactivable protein, but except that you don't need to pre-engineer your cells to use them. So here is an example. So in the cell line or the organoids showing here, there's no pre-engineered PA, GFP, or PA cherry. They just incubate the cells with a cell-permeable PA-JF549 dye. Once they do that and they found the cell that they want to isolate and do sequence on, they just shine light on the cell. And because the dye is cell-permeable, right, so the cell would now give you glow in red. And this would allow for follow-ups with the FACS sorting and sequencing events. So this works for both the single layer of cells, and also if you study organoid, which is like an organ-like group of cells that you can culture to study for a certain specific diseases. In this case, the authors use this method to photoactivate and isolate the whole group of cells in the same organoid, and then they can use sequencing to identify what is in this organoid. So this method is useful, which means that, okay, if you're in the middle of your PhD and suddenly you wanted to do targeted genomics, you don't need to go back and then construct your transgenic animal, which may take 3 more years, right? You can just use the dye and then proceed with the FACS sorting. Another method that has a great potential is that you can use a different type of molecule, which is a caged nucleotide. In this paper that's published in 2020, the authors call this method zip-seq. The zip means that they think that they're giving zip codes to the type of cells that they want to analyze. Okay, so this method uses caged nucleotides to do that. Basically, the caged nucleotides are nucleotides that has a bulky adduct. So this region, although it's single-stranded, it cannot interact with a different strand of nucleotide because this bulky thing that's interfering with any possible interaction. But this bulky thing here is going to be released upon light activation. Okay, so if you shine light on these things, then the bulky part will be released, and then now the single strand can interact with another molecule of DNA. So this method, the zip-seq, is designed for you to label multiple regions in a multicellular sample. For example, the authors can first incubate a sample with an antibody that's conjugated with the caged nucleotide. Right, so every cell, the antibody would interact with some cell surface protein, and so every cell would be coated with the antibody plus the caged nucleotide. And then the authors would go ahead and select for what they want to analyze in sequencing, and then they would draw a region of interest and then shine light in this region. Now the bulky adduct would be released, allowing the single-strand DNA to be hybridized with a second molecule that you add in the reaction. And the second molecule not only has a red part that can hybridize with the first molecule, but it also has a sequence that can later be read in a sequencer or the sequencing machine, which is what the authors call the zip code. Is the blue region. So you will add the blue zip code to this group of cells, and then the authors can repeat the same process to a second region of interest and add a different kind of zip code. In this case, the green zip code to the second kind of the cell. And then you can wash the cell and then dissociate them into single cells and go with single cell sequencing. Now, even though this zip code is added on the outside of the cell, there is a way to distinguish each kind of zip code after you do sequencing library preparation, which is preparing library based on what is in the nucleus, right? And this is achievable because the authors of that paper uses one of the newest technology for next generation sequencing called 10x genomics or 10x encapsulation. In this method, which I think is amazing, you basically make your sequencing library in a solution, like a tube, like this. In this solution, there are many, many small compartments. Each small compartment has, different barcodes that allows you to distinguish cells in different small compartments. And basically, the library preparation is made in these small compartments, which will result in, say, different single cell is going to have different barcodes that's recognizable later in the sequencing machine. So now, if you have the cell that's attached to a specific nucleotide, even though it's outside the nucleus, right? But if you can isolate the cells to, incubate in this one single cell gems, then this DNA sequence outside the nucleus would also be recognized by the same barcode, and then it would be recognized to be part of the cell. So even though this green or blue barcode never enters the cell, but in DNA library preparation, it would be counted as part of the same cell, which means after you send the whole thing for sequencing, you would know that, okay, the green barcode always comes together with these genomic DNA or mRNA molecules, and then the blue one goes with another cell. So in using this method, you can actually label more than one regions in the same population and allows us to analyze different things in the same sample simultaneously. The authors provided 3 different examples for that. First, they wanted to study wound healing, so this is a wound healing assay. They wanted to know what's the difference in expression pattern between the cells that's really close to the wound versus the cell that's kind of behind in the second line. So then they can draw the region of interest and then separate these regions and analyze the mRNA molecules in these 2 populations of cells. And the example in the middle is again the lymph node, except that in this case, instead of analyzing them separately, you can analyze the 2 different regions of the same lymph nodes and know what's the cell population there. And the example on the right is a mouse tumor. So you can use this method to study the tumor microenvironment and how it affects the cell, like expression pattern of individual cells. And now you'll be like, okay, how about if I want to analyze more than 2 different regions in the same tissues? You can still do that. So in the same paper, the authors already kind of designed that for you. Because now you're playing with the nucleotide, right? You can do endless hybridization of nucleotide and add more complexity or possibilities. And you can also combine this, a complex design of nucleotide addition versus different kinds of pattern of illumination. And easily, with combination of these 2 methods, you can create different regions, and that's differently defined by this surface nucleotide molecule. So in effect, you can sort them into different categories, and then you can analyze them in sequencer. All right, so in the last slide of today's class, I will only briefly share the last method, which is that this is a method that's focused on working with a fixed sample, right? So far we've been talking about live cell imaging or live cells, or imaging the cell in their physiological content, has neighbors, cell-to-cell communication. But how about if what I'm interested in is already a fixed sample? So the authors of this 2022 paper, they proposed a method in which that if you have a fixed sample with a permeable cells, and if the mRNA is still intact in those samples, you can perform a reverse transcription. So then from the RNA, you get cDNA. After you get the cDNA, which is still in situ, so it's still in the slice, right? You can mix the sample with a bunch of barcode that can hybridize with the cDNA. And the barcode can be photoactivated, which means that after you shine lights on the barcode, they will cross-link with the cDNA to make this funny shaped structure. After you have this structure, you can perform what they call the cross-junction synthesis. So you go from this funny shaped molecule to a linear fragment that has a barcode plus the cDNA. If you amplify these linear fragments enough, you can use this amplified barcoded cDNA to prepare your sequencing library and perform next generation sequencing. So even for a fixed sample, you can find ways to amplify the RNA and then prepare for next generation sequencing. All right, so hopefully I have convinced you that with the light illumination of the targeted cell followed by single cell sorting, you can have many different methods, and depends on what you're interested in, you can have different possibilities in your research, and hopefully maybe in several years we will say, okay, maybe you can actually use that in your own research project. And I will stop here and I can take any questions that you may have. Okay, anybody have questions? Any questions? No? All right, I'll be here for a few minutes more, and then if you have questions and you just don't want to ask in front of everybody, you can come to ask me. And if not, we will come back at 3:30, right? Okay, 3:20. We'll come back at 3:20, and then we'll do the student presentation. All right.